Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Brosnan, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speakers in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speakers your questions. Your questions in the question and answer windows will only be visible to myself and our speakers. So thank you for joining us today. It is now my pleasure to introduce our speakers, Jinjin and Senthil Kumar Kuppuswamy from WuXi Advanced Therapies.
Hello, everyone. Thanks so much for attending today's webinar. My name is Xing. I'm the Associate Principal Scientist and the Group Leader of Upstream Process Development at WuXi Advanced Therapies. Senthil, do you want to briefly introduce yourself?
Hello, everyone. I am Senthil, Associate Principal Scientist and Downstream Group Lead for Lenti 2.0 platform. Thank you.
Thanks, Senthil. So in this webinar, we will give you a general overview on our newly established Lentiviral vector platform. We will briefly introduce our company, client service, analytical and testing support, as well as some technical details on how we improve the titer and the yield with greater predictability in this new platform. WuXi Advanced Therapy is a global company across 2 countries and regions. For today, I will be mainly covering our US site that is comprised of 4 buildings with over 400,000 square feet located in the Navy Yard in Philadelphia. As you can see on this slide, we have dedicated building and space for process development, analytical and testing, clinical manufacturing, late phase, and commercial manufacturing for both cell and gene therapies. WuXi Advanced Therapies is a leading contract testing, development, and manufacturing organization, what we call CTDMO. We are a strategic GMP partner and offer integrated platforms to transform the production of cell, gene, and other advanced biopharmaceuticals. With a long history of customer service, we have become a global and fully integrated single-source partners to help accelerate your progress and time to market. Up to date, we have launched several platforms for AAV, Lenti, CAR-T, and beyond that can provide greater predictability and speed to market. We support many partners to accelerate their pipelines. For today, we will focusing on our latest advancement in Lenti platform. Up to date, we have supported 9 clients for Lentiviral vector productions, covering different therapeutic areas, such as myeloma, solid tumor, leukemia, and so on. This do help us accumulate valuable experience on Lenti process optimization and platform development. We all know that for cell and gene therapy market approval, developing a sustainable manufacturing process is a key. WuXi Advanced Therapies spend a great effort on building up strong PD capacity to support CMC activities throughout different phases of drug development, all the way from preclinical to final commercialization. We have experience working with lentivirus since 2015 and launched the world-class Lenti adherent platform in 2017. So this platform, version number 1, is developed with a completely closed process based on the hyperstack culture system. The production scale is 60 hyperstack 36, which can generate more than 200 liter material for downstream processing. We have completed a considerable number of GMP batches using this platform and definitely built up strong manufacturing capacity for Lenti production. However, it has to be admitted that we also see some technical challenges and a big room for improvement. That's why in early 2020, we initiated the development work for a new Lenti platform 2.0 in order to optimize the cell activity, transfection, condition, and the overall process flow. The aim is to upgrade the old Lenti platform with a robust, easy to use, and high producing and recovery process. On this slide, we briefly show the journey of upstream process development. At the beginning, the process optimization was conducted at small scale, primarily focusing on firstly promoting the cell growth and performance, like implement robust temperature control and shorten the pre-transfection culture duration. Secondly, enhance the viral productivity, such as transfection condition DOE, bring new plasmid system, and introduce expression booster. Then, the new cell culture process was verified at a large scale with more process control, like medium exchange post-transfection. Finally, we execute the technical transfer and onboarding the new platform in GMP suite. At this point, all the GMP documentation has been completed, and the new Lenti platform is fully ready for use to support client projects. In the next few slides, we will show you some experiment data, the improvement we achieved, and what the final GMP process looks like. In order to achieve high lentiviral vector titer, appropriate transfection condition is quite important. So this slide summarizes the DOE study we performed to optimize the cell density, DNA amount, PI ratio for transfection. Statistical analysis shows that maximizing the cell density and PI ratio within the design space can achieve the best titer. The data was further confirmed, and we can see the lentiviral vector productivity was significantly improved after optimization. Please be noted that we are not just focusing on these three factors. We also look at plasmid ratio and other important parameters. In addition, according to our experience, the optimal transfection condition vary a lot depending on different cell line and plasmids. So this kind of DOE study can definitely be built into the PD workflow if clients use their own. We also conduct the plasmid screening from different vendors and successfully identify oxygen plasmid can improve our lenti productivity by about 10-fold as compared to the control. Furthermore, after a number of verification runs in HyperStack 36, we saw that the new upstream process can further increase the titer by about 3-fold. So in total, Lenti 2.0 platform achieved more than 30-fold increase than before. With high Lenti productivity, the unit operation of upstream process can be much simplified, and downstream process can become more robust as well. This slide shows the overall upstream process flow at a GMP scale, a total of 18 days process. First of all, the cell wall is stored in HyperStack 12. Then, after 3 cell passages, the process jumped to 24 HyperStack 36 at production phase. 2 days after cell seeding, the transfection is conducted. And then 20 hours later, the fresh medium was exchanged. Finally, 3 days post the transfection, the supernatant is harvested for downstream processing. All the liquid handling is executed through the pre-designed tubing assembling, which can minimize the risk of contamination. In summary, we achieve a significant improvement of lenti productivity in our new platform by optimizing the plasmid and cell culture process. The new GMP process flow was also finalized with a robust and reproducible process control. So next, I'm going to pass over to my downstream colleague, Senthil. Thanks so much.
Thanks, Zin. That was a wonderful presentation. Here is a downstream part of the Lenti 2.0 platform. Here is the overview of our original Lenti 1.0 platform, which was launched in 2017. I would like to share the process improvements we made to increase the titer and recovery. Our original Lenti 1.0 platform process was 20 days long from vial thaw to harvest. The new upstream process improvements have shortened the overall time to 18 days. We perform media exchange on day 15, followed by addition of sodium butyrate. We continue to grow the cells following transfection for additional 3 days. This significantly improves the titer and provides much cleaner feed material for downstream process. On day 18, we begin our downstream process with harvest, followed by nucleus treatment, followed by clarification. Then we proceed to concentration and buffer exchange by ultrafiltration, diafiltration, which is followed by vial fill. All the downstream unit operations will be performed on the same day, as this improves the stability of the lentivirus. This slide shows the overview of Lenti 2.0 platform, which we just discussed. By having a very quick process with well-defined unit operations, we can ensure we can achieve the target titers and recovery with great predictability in GMP. Lentivirus, being an envelope virus, is susceptible to freeze-thaw stress. We evaluated various media which can give better cryoprotection against freeze-thaw stress. Our preliminary data shows significant increase in recovery and titer in optimum media and Tex-Max media when compared to our standard Lenti tris buffer system. In this slide, I would like to show the advantage of our platform process on the left to the custom process on the right. After the project has been initialized, we quickly proceed to predefined feasibility runs, which will allow us to know the expected titer and all the unit operations will be performed as desired. Feasibility run is followed by verification run, and the verification run will provide more information on the process performance at scale and the expected downstream process recovery, titers, and process residuals before we move to GMP manufacturing. Upon completion of the verification run, the process will be transferred to GMP manufacturing. This short approach gives our clients significant advantage compared to the custom process, where each step needs to be worked sequentially from tech transfer, bill of materials, batch records, process description, equipment list, and sampling plan before we even perform the verification run. This is followed by tech transfer to GMP and again followed by bill of material, procurement of raw materials, training of the manufacturing staff, and pilot run, engineering run under GMP manufacturing. With our platform approach, materials, training, and documentation are all in place, which will help clients significantly to reduce the cost as well as meet their timelines. For any new clients, we discuss the choice of plasmids and perform feasibility run and look for a target titer which our client desires. If the feasibility run meets the titers and product profile, we scale it up for verification run. If the process does not meet the client requirements, as with regarding to titer and recovery, as these are largely gene dependent, we perform predefined feasibility experiments before we proceed to the verification run. We are the only CDMO who provide end-to-end solutions for our clients. Our expertise covers the entire spectrum of services from early stage process development to release testing services. Here are some of the tests we routinely perform for monitoring residual and process impurities. Our analytical development team has qualified assays for various process residuals and impurities. Our analytical development teams not only support WuXi ATU clients, they also support clients in other divisions and CMOs. We have qualified assays for several gene and cell therapy products for both characterization studies and release testing. Here are some of the tests readily available for our Lenti platform clients. We have vector-specific release tests like titer assays, presence of transgene, flow assays, and transgene sequencing. We have a number of qualified assays for residual impurities, for determining residual DNA, host cell DNA, and residual benzonates. We provide number of release tests as listed, as well as for product safety. Our facilities are designed to meet all the quality and regulatory requirements for FDA, European Union, and other major regulatory agencies across the globe. We have a very good quality system in place, which are compliant with all the regulatory requirements and have programs to support the evolving regulatory requirements. We have a robust regulatory affairs team, which can help our clients for FDA meetings, pre-IND documentation, IND and BLA submissions. Our regulatory team can provide DMF authoring and submissions for US and Canada. They support pre-approval inspection as well as market authorization and maintenance. This concludes my presentation. Thank you for your time. Now I'm open to any questions on the floor. Thank you.
Okay. So the first question is, how robust is the HYPERStack system you have in your platform regarding cell growth and productivity as compared to small-scale adherent culture like T-Flask?
Yeah, this is a good question. So overall, we see the performance between HYPERStack and the small-scale T-Flask is pretty comparable in our platform. So our cell line can grow very fast in the HYPERStack system with doubling time less than 22 hours, I see. And for the Lenti Titer, yes, we see the Titer is slightly lower in HYPERStack 36 system, but it's not big off. It's still around the same level. Because we implement a number of process control methods, so we didn't see much technical challenge when we scale up and scale out the adherent process. Thank you.
Okay. Well, speaking of the titers, the next question is, what are the expected titers and yield on your Lenti 2.0 platform?
That's a good question. So titers and yield are largely gene dependent. On our model GFP gene, we get titers greater than 30% and recoveries of greater than 30% and titers around high E7. So safely we would say that we are around like 30%, and also the titers will be IE7 to E8 depending on the gene.
Okay, so it looks like we have time for just one more question. If we didn't get to your question, those questions will be passed on to our speakers. So feel free to continue to type in your questions, but we're just going to ask this last question now. What is the concentration factors you performed on your ultrafiltration step?
So our concentration factors are largely, we target the titer concentrations. We do sequentially, we take samples out to measure the concentrations before we proceed for further concentration steps. For the GFP, we are able to get E8 and greater than 30% at 30-fold concentration. So that also gives the flexibility for the clients. If they want higher titer, we can increase the concentration. On our Lenti 1.0 platform, we used to concentrate to 200-fold. On the since the cell line and our plasmids have improved, we no longer have to go such high concentration. We can go at 30 to 50-fold concentration, still be able to achieve higher titer and yields.
Okay, thanks, and thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.